Not like satellite tags. Satellite tags will give you a very fine precision on movement. Geolocators, they say, have a resolution of around 200 kilometers around any single point. And because they're light sensing, they're highly influenced by the equinox period. So you get a lot bigger error in location accuracy around the equinoxes, which is really annoying because equinox is in September when you want to know exactly where they're migrating to, and March when they're coming back home again. So that was a pain in the neck, but we got over it. Okay, so this is common Murres from Funk Island. Same thing, I just want to show you what they're doing throughout the year. Again, we have the data at the top. It goes a little slower, this one, because it's not as much data. They're not nearly as exciting. But it's interesting because those 3-4 individuals just hanging out on the Grand Bank for the whole time. For the whole, they sort of move off for a little bit. They're really centered in this area. This is going to be a repeating theme. Okay, so if you pool everybody together, take all those tracks, and you make a great big map of the, we're going to call this the, like a utilization distribution of what these birds are using for the whole year. The redder colors are higher densities of birds using those areas. So thick-billed Murres, again, from these 5 colonies, I just lump them all together. This is the wide distribution of ocean that they're using during the non-breeding season. And the common Murres from all 3 colonies are pretty much lumped in this area. If I had known all about tracking before I started my project, I may have tried to study birds like shearwaters or terns, which have these spectacular migrations across the globe. But because we didn't know very much about, Murres are very well studied, but it was appealing to think about, well, you know, we still don't know where they go in the winter. Next time I'll study shearwaters. Okay, so out of that big map I showed you with that big density distribution, I then made these hula hoops of where's 50% of the density of those populations centering? What's the core winter area? These are color-coded. So this is the Prince Leopold colony, and it's gray colored. I apologize for anyone who might be color challenged in the audience. This is the 50% core distribution of thick-billed Murres from Prince Leopold Island. Another example is the birds at the minarets, which are centered out here. So I did this for all the colonies. I did it for all the Murre colonies as well, which have a very high overlap. And then I calculated how much, sort of quantified how much overlap there was between all the different combinations of colonies. So let's look at that. We don't have to look at all these numbers, but I want to show you, so here's the colonies of thick-billed Murres, and how much, for example, the birds at Digges Island overlap with the birds from Coates Island. So those are these 2 right here. They're in the Hudson Bay, and they overlap the blue and the green. It's kind of hard to see, but let's just take my word for it. They're in the winter, core winter period, they overlap 50%, compared to birds from the Gannet Island and at Digges Island, they don't overlap at all. So this is a thick-billed Murre overlapping with a thick-billed Murre. What happens when you overlap them with the common Murres? And again, you see this is the area we're talking about. Common Murres and thick-billed Murres overlap anywhere between 0 and 40%. Thick-billed Murres and thick-billed Murres overlap somewhere between 0 and 50%. They both average out those 2 comparisons around 15% overlap in colonies. If you just look at how much do common Murres overlap with common Murres at those different colonies, it's very much higher. It's not a surprise, around 40%, but it's just a lot higher percent overlap. I'm going to take us one more step. So instead of a core colony distribution, let's lump all the birds from each different colony, as they're indicated by different colored lines. Let's look at the latitude, just plotting latitude of occurrence through time, so from September to May, and with the idea of let's try and look at what migration looks like. These are birds from Prince Leopold Island. And I want to kind of investigate how they move in terms of timing. Let's pick an arbitrary latitude, let's say 60 degrees latitude. Birds from Prince Leopold move slowly southward, and they don't cross that 60-degree latitude mark till sometime in November. Birds from the Minaret move southwards very quickly and crossed it early in the fall. Birds from Hudson and from Hudson Bay, the two Hudson Bay colonies, didn't leave Hudson Bay until December. These birds are moving southward throughout the season all the way to May, or April, sorry, when they then move back. So the point of this is that although the birds might be sharing particular parts of particular core areas or overlapping in habitat to some degree, they're moving through that habitat in different times of the year. So they might be not overlapping temporally. Again, you're sensing a theme. Common Murres are hanging out at the same latitude throughout the year. There's not a lot of variation in terms of when they're arriving, although these birds from the Gannett Islands, which are a little bit more northward, don't come down to that core wintering area till November. So there's less temporal differences. So I just want to review. So for the thick-billed Murres, we have very low, that 15% inter- and intra-specific core winter overlap, so between other colonies of thick-billed Murres and between the common Murres. It was more diverse movement strategy between the different colonies and sort of more variable movement times compared to common Murres, which have a high overlap, much more conservative movement strategy, and much less variable timing in terms of the year, throughout the year. I want to look at this in one more way before we move on from movement patterns. Let's look now at post-breeding, which is just a big lump of the fall distribution. And we're going to look at winter only, core winter, and then we're going to look at pre-breeding or sort of late winter, early spring time. So this is how the Murres are distributed post-breeding in the fall. You can see they're starting to move away from their colonies. Common Murres are doing the same. In winter, in the dead of winter, they've moved out of those northern bays. They're really spread throughout the Labrador Sea. Common Murres are totally concentrated. They concentrate even more in winter on the Grand Bank, Orphan Basin, and Georges Bank there. Thick-billed Murres continue to spread out in the late winter and spring, almost all the way to the mid-Atlantic ridge, which you can't see here, and there's no bathymetry, but they're really spreading out in the spring. Common Murres are still there on the Grand Bank. So there's a diverse seasonal movement strategy as well with the thick-billed Murres, so in terms of what they're doing in each season, and not so for the common Murres. Okay, so I'd like to now compare what we know, what we found out about the distribution, and look at that food web connection. So let's look at trophic position, and we're going to look at what happens again. Just reminding you that higher you go in the food chain, the higher trophic position you have, and that is why. So my methods for getting isotopic signatures from the birds, again, were to look at getting a blood sample, which tells you about the breeding season. The blood has a very high turnover, so anything incorporated in the blood is sort of recycled on a daily, weekly basis. So that's going to give you a signature of what's happening right at the breeding season. For feathers, what they're eating is incorporated, that signature is incorporated into their tissues when they molt, and that stays there until they molt again. So that signature does not change. So they molt their flight feathers in the fall, and so you get a post-breeding signal in the fall from the flight feathers, and they molt their body feathers sometime in the late winter or spring, and I took a feather here from the chest of the bird. That's the signature that's getting us the closest we can to winter distribution, end of winter. Okay, so you've seen my nitrogen graph before with those gulls. So here again, I'm just going to show you nitrogen. So this is increasing trophic position. All of the thick-billed Murre colonies are over here, and the common Murre colonies are over here. So it's just a spread. So this is showing you the trophic position of the different colonies with Prince Leopold foraging at a lower trophic level compared to this guy at Diggs Island, for example. This is, sorry, I didn't say, this is post-breeding. This is that fall signature. Let's look at the spring signature. The spring signature is much different among the thick-billed Murres, not much different among the common Murres. So we're seeing a drop in trophic position, particularly for the birds at Big Inlet Island. I want to stay at that point for a minute. Sorry to show you this graph again. This is the overlap thing, which I did core, winter, overlap. I want you to notice now this section is the thick-billed Murres from the Gannet Islands and how much they overlap with the common Murres from the three colonies. If you remember, in space, those are the ones that overlap the most of all the combinations. And the interesting thing is that during this period, during the period that we think is closest to that core winter period, their trophic position suggests that this is when they're most divergent in terms of the thick-billed Murres from the Gannet Islands are foraging at the lowest trophic position, and there's quite a difference between that and what the common Murres are doing. This suggests to me some things. Potentially that high amount of overlap might be influencing some ecological partitioning there. So again, thick-billed Murres having a very wide trophic position and diet partitioning potentially happening where there's a greater degree of overlap, spatial overlap. One of the more interesting questions is, for me, which I don't know the answer to, is those common Murres are hanging out in the same place. All the colonies are centered on the same place. They have a more narrow trophic position at a sort of high level. This really doesn't seem to be any diet partitioning at all happening. And that's kind of weird. Maybe they're all getting along. I don't know. Okay, let's pause there for a minute. Yes?
Does the age of the bird affect how far they travel?
That's a great question.
And what's the life expectancy of the Murre?
The Murre, I think, can live upwards of 20 years, or potentially more. Young birds tend to roam, just like in humans, young birds, teenage birds, tend to roam more widely. At least in terms of how many colonies they visit. There's this dispersal period early on in life, usually before they start breeding. So because these are late breeders, I can point to the adults and the chicks. Good time to ask this question. They have what's called delayed breeding. So they may not breed until 4 or 5 years old when they recruit to a particular colony. During that period, they're traveling all over. So there's a sense that they may travel more widely. The problem is we don't know the ages, mostly, of those birds that we track. And it's hard to, you can't put a transmitter on a chick either. These chicks, that's the age, this chick is about to fledge. And that's for the Murres, they fledge very young, and they finish their growth period at sea. So we can't track the chick, unfortunately. Not yet, anyway.
So you have no information on whether a chick left the colony and nested on a different colony, or the same colony that the parents did?
Only if the chick is banded. So there are lots of those banding programs, and if you opportunistically happen to band a bird and then for it to return, there is potential for finding that out, but it takes a lot of effort. You can imagine how many thousands you have to band before you start, before you start seeing them, particularly in a colony of a very large size.
So is there any evidence that the chick will return to the same colony or go to a different one?
Yes, there probably is, and there's. I can't tell you the number, but some proportion will recruit back to that colony, and others will end up elsewhere. Okay, so let's. What I take from all these differences in distribution and differences in trophic positioning is that likely between the species and between colonies, there's, to likely to be some differences in vulnerability to environmental risk, for example. So I'd like to walk you through a couple of kind of thought exercises. So we all know that birds encounter risk in the marine environment. It might be risk of hunting. These are the tur hunt you might be familiar with in Newfoundland. The Murres are still hunted for food. At certain times of the year, you have to get hunting permits and everything, but there's a risk to birds for being in nearshore areas for being hunted. Birds also may encounter, this is a map of offshore oil and gas licenses. There happen to be, oh, sorry, I just ruined my punchline. I thought this was a different map. There's a lot of offshore oil and gas activity. There's several platforms around Newfoundland, and those could potentially interact in a detrimental way with seabirds, particularly birds, which are oiled, which are highly susceptible to oil in the marine environment. You might get just chronic oil from shipping traffic, and this is a map of traffic in the region. And seabirds are incidentally caught, tangled up in fishing gear as well. So there's a variety of risks that they could encounter. So again, I want to think about now, how does the differences in distribution influence this vulnerability to risk? So again, let's just split up those colonies. This is a thick-billed Murre colony. That's its winter distribution. You're getting used to seeing these maps now. Let's just look at the differences when you separate out the colonies. So this is colony A, let's say. Colony B. Again, we're just looking at winter use. Colony C, D, E. So quite a difference in use, and also a difference in how much they're overlapping this nearshore area and a more sort of shelf or a more marine off-the-shelf area. People have tried to quantify the distribution of risk. So let's look at hunting, for example, the darker gray bar. Here's where hunting occurs in the coastal zone of Newfoundland. Here we have the incidence of where birds might encounter a risk of oil. These are all shipping lanes. And here we have, this is the corner of a fishing zone, so birds could potentially interact with the fishing areas in that particular area. So if you look at the spatial distribution of risk, and then you consider the spatial distribution of those colonies, you can do this experiment. You can do a kind of a thought exercise. Let's think about, of all these colonies, which birds are susceptible to hunting, for example. Hunting occurs in this nearshore zone. These are birds from the Gannet Islands. So they're only going to be influenced by hunting in this region, a little corner of the distribution. So let's just say we give them a little plus. Yes, they're influenced by hunting. We can do the same with all those other colonies. These birds here from Diggs Island, Coates Island, sorry, they're not intersecting that sort of hunting risky zone at all. So we give them a minus. And let's pretend we do that experiment and we rank everybody. It's just presence absence. It's just yes, they're influenced, no, they're not. We can do this. So sorry, before I keep going, does that make sense? Okay. So again, this is the map I was thinking about. There's offshore oil platforms here in the Orphan Basin and on the Grand Bank where there's active extraction of oil from the marine floor. There's also exploration licenses off the coast of Greenland, and as you know, they're sort of everywhere, depending on, there's some off here off the Scotian shelf as well. So let's do this little ranking exercise. What about those common Murres that are all overlapping the Orphan Basin and the corner of the Grand Bank? That's where those oil exploration activities are happening. So now we can say that all of the common Murres occurring from those three colonies have some kind of vulnerability to that risk in those areas based on what we know about their winter distribution. And we'll do it for everybody else. The guys up at Prince Leopold may be susceptible to activity up in the Davis Strait. Give them a little plus. So this is sort of an example of how we can move from distribution to risk. So if you can add different anthropogenic risks and figure out, in particular, what stands out is you start getting patterns. So maybe particular colonies